In this video, we're going to learn how to approach Biochem MCAT passages. This will include an overview of our initial reading strategy, as well as how to handle pathway problems. We'll begin with how to read passages. I like to break this into 3 simple steps. First, we're going to read, and our focus here is on the big picture. We're not going to reread, and if something goes over our head, then so be it. We're not going to worry about it. At the end of a paragraph, we're going to pause. And then lastly, we're going to think about our highlights. When we're thinking about our highlights here, this should really just be a landmark for what did they discuss, so that when we come back later, we will be able to efficiently and easily navigate the passage. At this stage, we're not going to worry about the details of particular pathways or data interpretation, because they may not end up asking questions about the things that you didn't understand. We can always come back to the passage later, figure out what information we need to answer the question in front of us, and at this point in time, it's often easier to understand what the passage was trying to tell us in the context of a question, because now we have an additional lens with which to view the passage. Now that we've overviewed our initial steps, let's go ahead and look at a worked example so you can get a sense of how this all comes together on an actual passage. This passage begins by saying spexin is a novel 14 amino acid peptide hormone found in high concentrations in the liver. This hormone plays a key role in the regulation of various metabolic processes and is implicated in metabolic alterations seen in obesity and diabetes. This first paragraph is simply introductory, and almost all MCAT passages are going to start like this. They're going to give you a couple sentences of very broad background information, and you really want to go ahead and pay attention to big picture, what is it that we're discussing? The rest of the passage will usually just give us more details about this. So in this case, what were we discussing? Well, apparently we're discussing spexin, which is this particular hormone. I would just highlight spexin in this case, and then they tell us a little bit later that it's key in regulating some metabolic processes. Now for me, I wouldn't highlight key in regulating various metabolic processes because I just care about knowing that spexin and the information around spexin is contained in this first paragraph and in the first couple sentences. From there, we'll go ahead and move on to the next paragraph. This next paragraph says, activation of the gallin receptor, a type of G protein coupled receptor GAL2 and GAL3 by spexin in the liver, results in downregulation of hepatic cholesterol 7-alpha hydroxylase 1, an enzyme that catalyzes the conversion of cholesterol into whatever that mess is. Through a series of steps, that mess is further converted into bile acids by the liver. This paragraph is specifically a pathway paragraph, and at this point in time, we don't actually care what the specific pathway is or what it is trying to tell us in the details of how it works. We can leave that till later once they ask us an actual question. Sometimes there'll be a really complicated pathway that's outlined in the passage, and they only end up asking one or sometimes even no questions about it. So we don't want to waste our time at this point in time going through and creating this nice little diagram of everything that's going on so that it can just sit on our whiteboard and ultimately not even go anywhere. So what would I take away then? Well, I want to capture that this is a pathway, and I want to also know what is the pathway involving. So I would just go ahead and highlight activation, since that sounds like a pathway style word, as well as gallin receptors. We're just talking about what happens when we activate gallin receptors, and a huge pathway then follows. Now that we have our highlights for our second paragraph, we'll go ahead and move on to the third paragraph. Here it says, to study the impact of spexin levels on obesity, researchers generated mice lacking the GAL2 and GAL3 receptors. Wild type and GAL- mice were fed a high-fat diet for 30 days, and their body weight was measured every 5 days for 30 days. The results are shown in figure 1. This is essentially a methods type paragraph, and for methods type paragraphs, again, I want to just go ahead and capture that this is a methods paragraph. In addition to capturing that this is a methods paragraph, I also want to figure out what is it that they were trying to study. So for me, I would just highlight study and the impact of spexin levels. So in this case, we're just trying to see what does spexin levels affect. Yes, we could highlight obesity if you really want, but this passage in this particular paragraph is so short that I'm probably not going to forget that piece of information, and the smaller my highlights are, the easier they're going to be to navigate later. Now that we've finished that, let's go ahead and move on to the next part of this passage, which is a graph. I don't like to interpret my graphs ahead of time because, again, especially if the passage has maybe 3 or 4 graphs, which is not super common, but it does happen, we have no idea if they're going to ask questions about every single graph that you see. In this case, we could waste a bunch of time making a bunch of interpretations that we may not remember. And so we're just going to go ahead and skip it. Now, when we skip a graph, we're not going to skip the entire thing wholesale. We're going to skip over to the figure caption instead. So in this case, I would just look past that big graph that's sitting in front of us and just read the figure caption, which says that this is figure 1, body weight of the GAL- and wild type mice fed a high-fat diet for 30 days. Since I want to know what this particular graph contains, I'll go ahead and pick a highlight from that figure caption that captures that idea. So apparently all it's going to tell me is the body weight of these 2 different mice, and we can figure out what exactly this data is telling us later, again, when a question actually asks us about it. And that's really it for our overall reading. If you want, you can take a moment to go ahead and summarize and look back through your highlights and see what am I taking away from this particular passage. In this case, I just reread my highlights and think about, okay, here we talked about this and this connects to this idea and we talked about this here and so on and so forth. So what are we talking about in this particular passage? Well, apparently we're talking about spexin, which is this hormone, and it has something to do with gallin receptors, and there's a whole pathway that's involved with that. They then wanted to study the impact of spexin levels on specifically body weight, and they show us some data about it. That's all I'm taking away at this point in time. You'll realize that I may not know that much about the passage, and that's totally okay. But if a question asks me about the gallin receptors of the pathway, I know exactly where to go, and I'm not going to be hunting from the very beginning, trying to get a grip on where the heck do I find information. This is why I prefer to read the passage first, rather than looking through my questions and then just trying to find the data. I feel like it leads you to be a little bit scattered, and so even though we aren't spending that much time on this passage really trying to understand it, I still think you should read it ahead of time rather than skipping straight to the questions and trying to pull out the relevant information. Now that we've read our passage, let's go ahead and talk about how we're going to approach our questions. I like to use a 4-step approach when answering MCAT questions, and I highly recommend that if you're new to MCAT style questions or you're struggling a little bit with your strategy, that every time you get to a question that's passage-based, you go through these 4 steps. They're not always going to be relevant, and eventually we'll get away from sticking to this really rigidly, and it should become a little bit more intuitive. But in the beginning, going through each step one by one helps you structure your approach and ultimately will lead to a very streamlined way of approaching these problems. Our first step is going to be to read the question and think about our content. Now, when I say read the question, I mean read the question and also the answer choices. We're not evaluating the answer choices at this point in time. We're simply skimming through them and looking for basic science words and things that we might know from all of that content studying that hopefully we've already done by the time that we're getting to passages. From there, we're also going to look at keywords, and keywords are specifically words that are familiar from the passage. These are typically not things that you're expected to know about outside of the context of the passage. So for example, if we're thinking about this passage, things like spexin would be a good keyword, as well as some of those abbreviations of the enzymes. Lastly, we're going to think about triangulation, and this is the process of finding information in the passage. Now, why do I call it triangulation instead of just go find stuff in the passage? Well, it's triangulation because when we triangulate things, we're trying to find the overlap and pinpoint a specific piece of information. In this case here, we're looking at the overlap between the content and the keywords. Once we've triangulated our relevant information, we're going to go ahead and solve the problem. Now, don't get me wrong, there is a lot of nuance to solving problems, and there are a lot of additional strategies that we can fill in here. But for the purposes of today's video, we're not going to really focus on the nuances of solve. I'm just going to show you how I would go about solving the particular problems that we see, and you can fill in a lot of the different gaps with other content videos that I've made that go through how to solve specific problems in the context of different content areas. All right, now that we've overviewed our 4 steps for solving problems, let's go ahead and see how we put this all together and answer some MCAT style questions. So this first question asks, spexin would be expected to weigh approximately, and then we have some Dalton and kilodalton values. The first thing that I'm going to be thinking about in this particular instance is the content. The thing that's coming up for me here would be protein, since I know about the weight of proteins, and I'm also thinking maybe something to do with amino acids, since I know that there is a correlation between the amount of amino acids, their average weight, and the weight of particular proteins. From there, I'm going to think about keywords. For me, the keyword would be spexin, since that's not something that I'm expected to know about outside of the context of the passage. When I go back to triangulate, I'm going to be thinking I need to go back to the very first part of this paragraph, since that's where they describe spexin, and I'm thinking I need to look for some information that contains amino acid or protein content areas. So when I read this first sentence, it says spexin is a novel 14 amino acid peptide hormone, blah, 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 blah. Then it goes on and talks about other things. What am I going to pull out from all of this? Well, the overlap between spexin and amino acid information would be that there were 14 amino acids. If I know that a single amino acid is 110 Daltons, then I can figure out what the weight of 14 amino acids would end up being, just by multiplying those two numbers together. So in this case here, that would be 14 times 110 Daltons. Looking through my answer choices, I can automatically eliminate both A and D. If we multiply 14 by 110, we're going to get a number that has a different integer value than 14. Now looking between my two answer choices of B and C, I have either 154 Daltons or 1.54 kilodaltons. Let's just think about this here for a moment before we do any math, if we even need to. If one amino acid is 110 Daltons, then 14 amino acids has to be way more than 154 Daltons. So in this particular case, the correct answer must be C. And oftentimes, when I'm approaching math, I'm first going to see if I can solve it without having to actually crunch the numbers. Now that we've seen one instance of this particular strategy, let's keep going and continue to reinforce this idea and the steps and how we go about solving them. So this next question asks, the organ in which CYP7A1 acts also serves to, and then we're given some different answer choices here. As I scan through my answer choices, I see that they're talking about regulating osmotic pressure and something about protein synthesis, storing bile to aid in the digestion of lipids, secreting insulin in order to import glucose into cells, and producing specialized antibodies that can attack and kill bacterial pathogens. If this was asking me about an organ and then gives me some functions, I'm just thinking these specific functions must be tied to a particular organ. So in this case here, my content information would be maybe digestion, since a lot of it had to be around the digestive system and specifically organs of digestion. There is also that kind of weird piece about antibodies. So, your content doesn't have to be perfect. That's probably good enough. Well, what about the next piece? In this particular case, we're going to go ahead and think about our keywords. And our keywords in this case for me would be CYP7A1. That's really specific to the passage. So I'm going to look for the overlap between organ, CYP7A1, and we'll go from there. So here it says that activation of the gal-renin receptors, a type of G protein coupled receptor gal-2 and gal-3 by spexin in the liver, results in downregulation of hepatic cholesterol, blah, 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 that enzyme that we were looking at. So it sounds like what we're describing here is the liver. So what I need to figure out is, does A describe a function of the liver? Does B describe a function of the liver? Does C describe the function of the liver? Or does D describe the function of the liver? So let's go ahead and think about A first here. Regulate osmotic pressure in the blood through specialized protein synthesis. So this is just talking about the creation of albumin, and that is something that the liver does. So that would be the correct answer in this case. Well, what about the rest of them? B says store bile to aid in the digestion of lipids. The liver creates bile, but it doesn't store it. Storage occurs in the gallbladder, so B would be referring to the gallbladder, making that incorrect as well. C says secrete insulin in order to import glucose into cells. The pancreas would be secreting insulin through the beta islet cells, so that's also wrong since it's not describing the liver. Lastly, we have produce specialized antibodies that can attack and kill bacterial pathogens. This to me doesn't seem to be describing an organ, but rather a cell, specifically plasma cells, since they produce antibodies. I don't really know what to think about that specialized antibodies piece, so this is probably just kind of a weird distractor answer that doesn't really pertain to this particular question, and so we can also eliminate it in this case because it doesn't pertain to the liver. Now let's move on to our next question. This one here asks, which of the following mice would be least likely to gain weight when fed a high-fat diet? As we scan through our answer choices, we can see that they discuss microRNA in the spexin transcript, drugs that downregulate adenylate cyclase, agonists of gal-2 and gal-3, and allosteric activators of CYP7A1. As I begin to think about this, I want to identify any of my content pieces of information at this point in time, and ultimately I don't see anything that seems to fit into one box. In this case, I'm just going to go through and think about words that I've maybe seen before or that stick out to me. So for me, that would be microRNA, adenylate cyclase, agonist, and allosteric activator. As I begin to look at everything that I've highlighted at this point in time, I'm getting the sense that this has something to do with signaling, since we get this idea of agonist, activator, adenylate cyclase. Don't know necessarily what to think about the microRNA, but that's okay. And from here, we'll go ahead and move on to picking out our keywords. There are a lot of keywords in this case, and we want to go ahead and just pick out as many as we can. For me, that would be high-fat diet, or HFD, spexin, the gal-2, gal-3 receptors, and CYP7A1. Now that we have our general content information as well as our keywords, we'll begin to triangulate this information in the passage. Since there are a lot of keywords, I want to just go ahead and look back to the passage and see where they're showing up. I will always start with the question stem, and we'll begin by looking for HFD, and then we'll look for things that showed up in the answer choices, and then we can pick and decide what from there is actually going to be relevant. Here, HFD shows up twice, once in the method section and once in the figure caption. Always make sure that you're looking at your figure captions when you're looking for these keywords. They like to sneak them in there, and you can miss out on some important information if you don't pick up on that. Now let's look for the remainder of our keywords. That would be spexin, gal-2, gal-3, CYP7A1, and in this case, they're going to show up both in the methods and the pathway section. Since our keywords show up in a lot of areas in this particular passage, we need to think about what is the core of this particular question before we just go and grab information. So let's go ahead and reread it again and hone in on what is actually important and what can wait. So as I look at this, it says, which of the following mice would be least likely to gain weight when fed a high-fat diet? So the first thing that I would be thinking is, are any of these areas describing weight gain? And do they have something to do with high-fat diet? So I'll start by looking there, and I know that they were measuring the weight in this particular graph. So I would look at this graph, and that is where I see figure 1 caption says that this is the body weight of the gal minus and the wild-type mice fed a high-fat diet for 30 days. So since I'm looking for something that's going to be least likely to gain weight, I would begin by looking at these two different conditions here, describing what they are on the basis of the methods, and then just labeling them as either they gained more weight or they gained less weight. As I look through the methods, it says up here that we had this wild-type and the gal minus, and that gal minus lacked the gal-2 and gal-3 receptors. If we then cross-reference the gal minus condition with a graph, we can see here that if you have low gal, or in this case no gal, then you're going to gain more weight. But if you're the control, that would mean that you have higher gal function and you would gain less weight. That's a really good indication of the right answer here. Since we were looking for a mouse that would be least likely to gain weight, we should find something that has high gal function, and that's exactly what C says. A mice in which a gal-2 or gal-3 agonist was administered, since agonists increase the function of that gal-renin receptor, then it would presumably lead it to gain less weight, making C correct. Even though it isn't necessary to eliminate the remainder of the answer choices in this particular case, I want to go ahead and do this so that you can see the strategy for integrating the pathway with the data and how ultimately that pathway is going to show how all of these answer choices are incorrect. In this case, we're going to need to jump back to the pathway portion of this graph, and you might wonder, how do I know that this is the case? If we think about the way that this particular question is being phrased, they're talking about downregulating or upregulating a lot of these different elements that were in our pathway. If you see language around that, there's a really good chance that this is a pathway problem, and charting out the full pathway is going to ultimately be important. If we look at our keywords, we can also see that a lot of these important keywords were showing up in our pathway, and for me, the really big indicator that we need pathway information is CYP7A1. That doesn't ever show up in the method section, and so that would mean that the pathway must be important in terms of answering or at least eliminating the remainder of these particular answer choices here. To get started, we'll just go ahead and reread this particular pathway and then begin to chart it out in a flowchart method. So here it says that activation of the gallin receptor, a type of G protein coupled receptor GAL2 and GAL3 by spexin in the liver, results in something. So the first thing that we want to know is what was activating the gallin receptor, and here they tell us that is spexin. So if we have spectin, then we're going to see an increase in the amount of gallin receptor activation. Well, what does this lead to next? Well, this apparently results in the downregulation of hepatic cholesterol 7 alpha hydroxylase 1, which is CYP7A1, and therefore we can just go ahead and draw a little downregulation of this. Now, we could continue with this particular pathway and chart out the entire thing, but in this case, we don't need to because we've captured all of our keywords in our pathway. We can stop. We should have enough information to be able to answer this question, so we don't need to understand the rest of it and all those other pieces that have to do with cholesterol or bile acids. If another question happens to ask about that later, we can always just add on to the pathway that we've already created. Now that we have our pathway, we want to go ahead and figure out how each answer choice is going to impact this particular pathway. And the best way to begin to think about this is this increasing or decreasing one of the elements within this pathway? So for example, this first answer says that mice were injected with a microRNA that hybridizes to the spexin transcript. MicroRNAs are going to hybridize to specific transcripts and will down-regulate the amount of them. So what would this lead to? Well, this would lead to a decrease in spexin. If we had a decrease in spexin, we would see a decrease in gal function, and we know that if we have lower gal function based on the graphs, then we would gain weight. Therefore, A would lead to a mouse that would be more likely to gain weight rather than one that would be least likely to gain weight. Therefore, it must be incorrect. We will now simply repeat this process with answer choices B and D, and we'll see again why these particular answer choices are wrong. B says that mice administered a drug that downregulates the normal function of adenylate cyclase. Let's go ahead and skip this for right now because it's not directly in our pathway, and we'll look for some information that might relate to adenylate cyclase, but we don't really have the information pulled out of our passage at this point in time, so we'll just move right along to D. Answer choice D says that mice were given an allosteric activator of CYP7A1, and now we need to connect this to body weight. This is the opposite of what we see in our pathway here. Normally we see an increase in gal leading to a downregulation or a decrease in CYP7A1. So that would suggest to me that we would see a decrease in gallin receptor leading to an increase in CYP7A1, which would fit with allosteric activation of that particular enzyme. It's reasonable to conclude that if normally high gal function would lead to low CYP7A1 function, then presumably that would also lead to less body weight. Since we've flipped this particular relationship, we should expect high CYP7A1 function to be associated with a gain of body weight, making D incorrect as well. Now let's jump back to answer choice B and think about adenylate cyclase a little bit more. Chances are we need a little bit more passage information because at present I don't see a way that adenylate cyclase actually connects to what we're looking at here. Now that doesn't necessarily mean that you can automatically eliminate this type of answer choice because it's not related to the graph. They can disguise the relationship here, and we're going to actually see a case of this. If we go back and we look at this pathway, it says activation of the gallin receptor, a type of G protein coupled receptor. Well, G protein coupled receptors generally upregulate adenylate cyclase. Now, in this case, we don't necessarily know that's true, but it doesn't matter because it says that B is mice administered a drug that downregulates the normal function of adenylate cyclase. So whatever the normal function of adenylate cyclase is in this case, it's being knocked out. That's basically the same thing as saying knocking out gal function, and we know that if we have less gal function, then we're going to see a gain of body weight, making B incorrect as well, since it would lead to a mouse that would be most likely to gain weight rather than one that is least likely to gain weight. Now let's look at our last question here. This one says, an siRNA that hybridizes the spexin transcript was introduced into wild-type mice. Which of the following would you expect to see in this mouse population? Underactive CYP7A1 enzymes, mice with low body weights, decreased levels of liver cholesterol, or low bile acid production. Again, we'll start the same way we've started all of the rest of our questions, which is by looking for content information. As I scan through here, there are a lot of keywords, but the only thing that sticks out to me as being particularly content forward would be siRNA. From there, I'll go ahead and pick out my keywords. Remember, these things are going to be passage specific. For me, that would be spexin, CYP7A1, liver cholesterol, and bile acid production. Since I remember body weight being important in the last question, I would also highlight that since I know this graph will tell me something about the connection between body weight, mice, and spexin. Now we'll repeat the same process as we did last time, finding our keywords in this passage, and we're going to see the exact same pattern pop up in this particular case here. We're going to see a lot of these keywords in the method section as well as in the pathway problem and also coming up in the graph. In this case, the pathway is going to be more important because we have a lot more of those keywords coming up. In this case, we go a little bit further than we did last time. While we do include things like spexin and CYP7A1, we also have liver cholesterol and bile acid production. So let's begin recharting out our pathway and go all the way to the end of it. So again, it says here that activation of the gallina receptor, a type of G protein coupled receptor, is occurring by spexin. So we'll start with spexin and then draw an arrow and up to the gallina receptor since it's upregulating it. What does that lead to? Well, this apparently results in the downregulation of CYP7A1. So we just draw a down arrow for CYP7A1. From here, we'll continue to move forward and see what happens next. Apparently, this enzyme is supposed to catalyze the conversion of cholesterol into this big mess here. And so I would go ahead and draw this out as a particular reaction. So cholesterol will go to, and I'm just going to abbreviate it here as 7-alpha. At this point in time, I want to think about what is the relationship between this reaction and what I see in my pathway. Well, normally this enzyme is supposed to catalyze and make this reaction happen a little bit more quickly. Now, in this particular instance, that isn't happening. We're actually going to be downregulating this enzyme, which would block or at least slow down the conversion. So I'll just draw a little X here showing that cholesterol is not going to be converted into 7-alpha hydroxy, whatever nonsense. I'll also draw a little down arrow next to that so I know that that's being decreased, and then we'll continue on with the rest of our passage. Here it also says that through a series of steps, that whole mess again is converted into bile acids by the liver. So presumably, if we have low levels of 7-alpha, then that would lead to low levels of bile acids as well, since we don't have the precursor molecules we need to actually go and produce those. Now we can begin to actually answer this question. And in this case here, it's talking about an siRNA, and that's going to hybridize to the spexin transcript. This is very similar to a microRNA. siRNA will also downregulate the amount of a transcript that actually gets translated. And so all this is really saying is that we're going to see a decrease in spexin. From here, we can simply go through our particular pathway and chart out what would happen if we decrease spexin at each particular piece in time. And then we should be able to find our answer choice on the basis of this. If we do this, we're going to need to change a lot of our arrows because remember, this is talking about the presence of spexin. So we'll start by drawing a down arrow saying we're not going to have spexin, which means that we need to flip the gallinin receptor arrow and say we're not going to have gallinin activation. If we don't have gallinin activation, then the CYP7A1 enzyme will not be downregulated, and so we can go ahead and put an up arrow there. From there, that means that the cholesterol 2, 7-alpha hydroxy, blah, 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 must be going forward. So we'll go ahead and erase our original X that was going through that arrow and redraw it. This must mean that we're going to have plenty of that precursor molecule that leads to bile acids around, and we should see high amounts of bile acids if we have reduced the amount of spexin that was present in the beginning. Now we'll just go through and match up our answer choices and see if they agree with our new altered pathway or if they say something that is opposite of it. So for example, answer choice A says that we would have underactive CYP7A1 enzymes, but if we look at our chart, we can see that we have an up arrow next to the CYP7A1. That means that we would have overactive enzymes, making A incorrect here. Since our pathway doesn't include body weights, we'll go ahead and skip B and we can consider it a little bit later. Let's go ahead and move on to C since liver cholesterol was a part of this particular pathway. Here we have to think about what's going on and see, would we have decreased levels of liver cholesterol? Well, if we have high amounts of CYP7A1, which is what we would get if we reduce the amount of spexin levels, that means that we're converting more cholesterol to 7-alpha hydroxy, blah, 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 blah. That would mean that if we have more cholesterol conversion, then we would have less cholesterol in our liver. That means C would be the correct answer choice here. And then now from here, we can go ahead and move on to D and see why this answer choice would also be incorrect. In this case here, we can see that if we have low amounts of spexin, at the very end of our pathway, we should have high amounts of bile acids being produced. D says that we would have low amounts, making it incorrect because it should have said high. Lastly, we can go ahead and look at B, and for this, we'll jump back to our graph. If we investigate our graph, we just need to kind of connect this back to the gallinin receptor, because remember, the graph was connecting body weight with the gal receptor, and we have gal receptors in our pathway. In this particular instance here, we can see that we have low gal. Low gal would correspond with the knockout in this particular case, or that dotted line, the gal minus, and we can see that the gal minus ends up having a higher body weight when we compare it to the control. So we should expect to see mice with higher body weights, not mice with lower body weights, if we decrease the amount of spexin that is present in this particular situation. Hopefully this video has given you a good idea of how to attack BB passages on the MCAT, and ultimately the strategies I've discussed here can be applied to both the CP and Psych-Soc sections of the exam as well. If you found this content helpful, go ahead and let me know down in the comments section if you want to see more of this type of video. And as always, make sure that you go ahead and like and subscribe for more helpful MCAT tips, and share this video with anybody else who might be taking the test.